Hey everybody, it's now time to learn about the cell cycle. We will start today with looking at mitosis. We're going to figure out how cells are produced, how cells produce identical cells through mitosis for growth, development, and repair. The other two bullet points listed here will come a little bit later in this unit. Before we jump into mitosis, we want to have one little side note about prokaryotes. Remember, prokaryotes do not have a nucleus, so they don't need to go through the detailed process of mitosis. The way they produce is a process called binary fission. This is very straightforward, very simple. So let's draw a little picture of that. So if we have a bacteria cell, of course it will be unicellular. It also has one circular chromosome. In order to make two cells, the first thing that we would have to do is to make a copy of that chromosome. If you don't make a copy of it, then each cell will not get identical DNA. Now, once we have a cell with two chromosomes in it, all that happens is that cell will pinch in half and divide into two. These two cells are identical to each other, and they have the same DNA as the cell in the beginning. The only difference is that they're just smaller because they just divided. Okay, now everything else we talk about is only going to relate to eukaryotes. And when we talk about eukaryotes here, we're talking about plants, animals, fungus, and all types of protists. The cell cycle consists of three parts: interphase, mitosis, and cytokinesis. If you look at this picture right here, if a certain cell's life is 16 hours, well, most of it, a full 15 hours, would be in interphase. Mitosis is just a short little piece whenever the cell is going to divide in half to make two new identical cells. Interphase, there are three parts to it. They are referred to as G1, S, and G2. G stands for gap. So it's just the time in between other events. G1, just normal cell activities, and the cell is growing and getting larger. During the S phase, S stands for synthesis, which means to make, DNA is copied. If the DNA is not copied, well, we can't have two new identical cells made. And then G2, we have normal cell activities and growth, as well as preparing for cell division. Now, some cells are not actively dividing. This would include muscle cells and liver cells, nerve cells. And if they are not actively dividing, they are something called G0. Some cells, like your liver cells, can leave the G0 phase and then go back into the normal cell cycle. Other cells, like your nerve cells, are in a permanent G0, meaning that they will never divide. And here's a nice little Amoeba Sisters animation of how this works. So we have, G2, getting a bit larger, preparing for division, splitting apart. Then we have two new cells. The cell gets larger. We enter the S phase where the DNA is copied, back to G2, M, and G1. Okay, for more details. In interphase, if we look at the basic idea of here, we would have a centriole, and then the DNA is in the form of chromatin. This is loose DNA. The nucleus is intact. We have a nucleolus. And don't forget, we also have all the other cell parts here. We just look at the nucleus and the DNA to make things simple here. Okay, so for cell division, consists of two different parts that kind of happen at the same time. We have mitosis, which is division of the nucleus. Cytokinesis is when the cytoplasm or the cell itself actually divides in half. If you had mitosis occur but no cytokinesis, you would have one cell that has more than one nuclei. There are some cells that do this, like fungal cells and muscle cells. It is a form of asexual reproduction because we're making two identical cells. Some organisms will use this for actually making a new organism, so unicellular eukaryotes. But normally it's going to occur for growth, so to get bigger, development, and then repair. So if your skin is cut, it grows back by mitosis. Here's some pictures of actual plant and animal mitosis. We're going to draw some nice little diagrams, but it's important to keep in mind that actual life is not quite that neat. Okay, here's a preview of the stages of mitosis. Okay, let's move on. So the stages of mitosis. Remember PMAT. P will be prophase. During prophase, the cell prepares for mitosis. This includes chromatins condensing to form chromosomes. This makes it easier for the cell to evenly divide its DNA. The nuclear membrane is going to disintegrate in order for the chromosomes to move around. If the nuclear membrane did not break down, well, the spindle fibers could not attach to the chromosomes and move them to opposite ends. Notice that the spindle fibers are attached to the centrioles on one end, and they're just protein fibers that shoot out that will eventually attach to the chromosome. And then we have metaphase. Metaphase is when the chromosomes meet in the middle. The spindle fibers will move the chromosomes, line them up all nice and neat so we can evenly divide the DNA. This brings us to anaphase, when the chromosomes move apart. If we look at this first chromosome, we remember that it used to be connected, now it is separated. This strand of DNA is identical to this strand of DNA. This means that the cell that will form on the left is going to have an exact copy of the DNA from the cell that's going to form on the right. And again, the spindle fibers are what actually move the chromosomes around in the cell. Without without spindle fibers, we would just have our chromosomes floating around and just chaos. Then we have telophase and cytokinesis, when the cell is going to tear apart. The nuclear membrane on both sides will reform around the chromosomes. The chromosomes will also uncondense to go back to chromatin. We're looking at an animal cell right now because it does not have a cell wall. In animal cells, what will happen is a cell membrane will pinch in and separate the two cells. This place it pinches in is called the cleavage furrow. Plant cell cytokinesis just has one additional feature. Since it has a cell wall, it can't pinch in. Instead, we're just going to grow a new cell plate in the middle of the cell to form the new cell wall. Every other feature of mitosis is the same, with one small detail. Plant cells do not have centrioles, but they still have an area that will organize the spindle fibers. One last important point is that your cells are very smart. They know what they're doing, and there is a huge amount of control in how cells know what to do and when they need to do that. All of this control, by the way, comes from the information that is in the DNA, and the DNA will make those proteins that will actually do what needs to happen. We talk about the cell cycle. There are 3 important checkpoints that determine whether a cell will continue to divide. It's shown on this diagram right here. So in order to go into the S phase, the cell must pass the G1 checkpoint. In other words, it checks to make sure everything is correct to keep on going through the cell cycle. If everything is not correct, well, that means everything will pause, the cell will fix itself, or it might lyse or burst. There are 2 main genes that will control how the cell works. This is the tumor suppressor genes As the name suggests, it's going to suppress a tumor. We also have proto-oncogenes that are going to promote or tell the cell when to start dividing. You may have heard of the word oncologist or oncology, and that refers to a doctor or the study of cancer. Proto-oncogenes, these are the genes that everybody has that, if they are mutated, can lead to cancer. By the way, mutated tumor suppressor genes can also lead to cancer. So mutations in any of these genes, any variety of mutations, can lead the cell to divide uncontrollably. What is supposed to happen is that your body has a whole system of checkpoints. You have an immune system that should take care of these cells. But of course, sometimes these cells are dividing uncontrollably, go unchecked, and this can lead to tumors and cancer. Remember, we're talking about lots of complex genes and how they work together. So when we look at what a cancer cell is, every cancer cell is going to be caused by a slightly different mutation in a different part of your body, which leads to the fact that it's very difficult to figure out how to treat and cure cancer. At this point, you should be able to explain how cells produce identical cells through mitosis for growth, development, and repair. As you go back and review over this, really pay attention to the mechanism cells use to make sure that each new cell receives an identical copy of each original chromosome.